Welcome, fellow scientists, researchers, and toxinology enthusiasts, to this explainer. Today, we're diving into something absolutely fundamental to our field, and honestly, we're kind of ripping up the rulebook. For decades, we've relied on this phenomenological classification of snake venom toxins. You know the drill. We name them based on the tissues they appear to damage. But as we're going to see, this framework is completely obsolete. In this explainer, we're exploring the urgent paradigm shift toward a precise, structural, mechanistic framework that actually reflects molecular reality.
Okay, so here's our roadmap. We'll start with the tissue damage burden, then get into rethinking taxonomy, and dissect both direct cytotoxicity and indirect ECM degradation. Finally, we'll wrap up with clinical pathophysiology and the really exciting stuff, novel therapeutic inhibitors. Let's get to it.
I want to hook you right away with this question. Is our entire classification system fundamentally flawed? Well, think back to the 1940s. Scientists first described a cobra venom toxin that caused cardiac failure, and understandably, dubbed it as a cardiotoxin. But it turns out that was a massive historical error. Later studies prove the primary effect of these toxins is actually at the membrane level, causing cytolysis across various cell types, definitely not just the heart. So naming toxins based solely on the first tissue they appeared to damage, rather than their true biochemical mechanisms, that has created a massive blind spot in our scientific vocabulary.
Part 1. The tissue damage burden. Let's take a quick look at the global clinical reality to understand why this scientific shift is so urgently needed.
400,000. That is a staggering number. Every single year, there are conservatively 400,000 cases of severe morbidity resulting from snake bites. Now, while neurotoxicity and hemotoxicity often dominate the mortality statistics, it's actually the tissue-damaging toxins that cause this massive burden of lifelong disability. We're talking about permanent muscle tissue loss, contractures, acute kidney injury, and severe disfigurement. And this disproportionately impacts the daily lives and socioeconomic stability of victims who, let's remember, are often young agricultural workers.
And the disconnect in our approach is just stark. You have these medically important viparade and elapid species, like the jararaca and the spitting cobra, causing immense physical destruction. We're talking severe swelling, blistering, and deep tissue necrosis. Yet our current medical vocabulary loosely throws around terms like myotoxin, which completely fails to accurately describe the complex molecular warfare actually happening in those tissues.
Which brings us to Part 2, Rethinking Venom Taxonomy. It's time to evolve our vocabulary.
Take that outdated term myotoxin. A single myotoxin could be a phospholipase A2, or it could be a metalloproteinase. Sure, both damage muscle tissue, but they do so via entirely different molecular mechanisms. Organ-based naming is just obsolete. By shifting to a mechanistic classification, terms like cardiotoxin become cytotoxic three-finger toxins, and myotoxins are appropriately specified as cytotoxic PLA2s. This tells us exactly what the protein family is, and crucially, how it behaves at the cellular level.
When you look at how these venom families overlap, the sheer complexity is wild. True tissue-damaging toxins span multiple protein families and even intersect with neurotoxins and hemotoxins. Because of this complexity, it is scientifically imperative that we divide them into two distinct categories: direct cytotoxins that aggressively compromise cell viability, and indirect extracellular matrix degraders that essentially destroy the cell's surrounding environment.
All right, Part 3, Direct Cytotoxic Mechanisms. We're starting our deep dive at the molecular level with that first category, the true direct cytotoxins that aggressively target cell membrane integrity.
These three primary actors destroy cellular viability directly. Cytotoxic three-finger toxins and PLA2s are notorious for this, often working in synergistic complexes to potentiate their effects, which is a truly fascinating evolutionary adaptation seen in spitting cobras. But as scientists, we have to admit a gap in our knowledge here. Despite decades of research, we still don't fully understand the exact mechanism of three-finger toxin pore formation, or exactly how they oligomerize on the cell membrane. There's just so much left to discover.
Let's look at the actual mechanics. PLA2s completely destabilize the lipid bilayer. They do this through phospholipid hydrolysis, which destroys the membrane potential and causes this massive uncontrolled influx of calcium right into the cell. That leads to mitochondrial dysfunction and ultimately necrosis. Meanwhile, the highly positively charged beta-defensin-like toxins take a totally different route. They penetrate the membrane via endocytosis, accumulate in the lysosomes, and basically rupture them from the inside out, triggering apoptosis.
Moving on to Part 4, Indirect Extracellular Matrix Degradation. Let's look at the toxins that destroy the structural support holding cells together, rather than attacking the cell itself.
Enter the snake venom metalloproteases, or SVMPs. These are zinc-dependent enzymes. Instead of lysing the cell membrane directly, they hydrolyze the crucial structural proteins of the basement membrane, things like collagen type 4 and laminin. By destroying this interstitial matrix, these SVMPs cause profound secondary damage. They literally pull the rug out from under capillary endothelial cells, disrupting microvessels and ultimately leading to severe ischemic necrosis, because the blood supply is completely cut off.
In vitro, we see some truly fascinating behaviors with this. When SVMPs degrade the extracellular matrix, cells detach from their substrate. This loss of adhesion triggers a very specific form of apoptosis called anochis. Additionally, we see L-amino acid oxidases, or LAOs, acting as another indirect mechanism. They catalyze oxidative deamination, which produces incredibly high levels of reactive oxygen species, primarily hydrogen peroxide. This denatures enzymes, damages DNA, and triggers apoptosis.
So, Part 5, Clinical Pathophysiology in Vivo. We understand the molecular disruptions now, but how does all this microscopic chaos translate to a human patient during an actual bite?
Systemically mapping these toxins reveals a pretty grim picture. When you inject a venom high in SVMPs, that zinc-dependent catalytic degradation of the basement membranes doesn't just stay local. It directly leads to the weakening of capillary walls, massive extravasation, systemic hemorrhage, and ultimately cardiovascular shock and multiple organ failure. The molecular mechanism directly drives the macro-level systemic collapse.
Tracking the cellular mechanism directly dictates the clinical outcome. Cytotoxic PLA2s target skeletal muscle, leading to systemic rhabdomyolysis. This massive breakdown of muscle releases myoglobin into the bloodstream, which in turn drives severe acute kidney injury. Meanwhile, hyaluronidases act as spreading factors, degrading hyaluronic acid to disorganize the matrix and allow other toxins to spread even faster. This right here is exactly why mechanistic taxonomy is so critical. It maps the exact path to the pathology.
Which leads us to our final section, Part 6, Novel Therapeutic Inhibitors. Understanding this exact biochemistry isn't just for textbooks. It unlocks our ultimate goal in toxicology, targeted, rational drug design to stop necrosis in its tracks.
This new taxonomy builds an incredibly compelling case for targeted drug design. Traditional antivenoms rely on large, animal-derived antibodies. They're often delayed and have notoriously poor tissue penetration, making them largely ineffective at halting rapid local necrosis. But by identifying precise enzymatic active sites, we are paving the way for small molecule inhibitors. These are drugs that can reach the local bite site rapidly, penetrate the tissues deeply, and shut down the destructive enzymes immediately.
The sheer potential of repurposing specific enzyme inhibitors is incredible. Take Verisplatin, an indole originally developed for inflammatory diseases. It directly and powerfully inhibits the PLA2 active site. Then look at the SVMP inhibitors. Vatimastat directly binds the zinc ion within the catalytic pocket of the metalloproteinase, while drugs like dimercaprol work by chelating the zinc moiety entirely. By blocking the catalytic pathways, we can neutralize the venom's most destructive properties at the source.
Which brings us to the most exciting frontier in snakebite research. If we can successfully inhibit these precise molecular mechanisms using oral, rapid-acting small molecules, can we halt local necrosis right there in the field before the patient even reaches a hospital? I want to leave you with this final thought. Adopting a mechanistic taxonomy isn't just an academic exercise in pedantics. It is the necessary blueprint, the direct path to pre-hospital therapeutics that could save hundreds of thousands of limbs, livelihoods, and lives every single year. So what role will you play in this new paradigm? Thank you for joining me in exploring the science behind the